You lie still, and in the quiet of your breathing, you begin to sense something subtle. Beneath your skin, beneath the steady rhythm of your heart, beneath even your thoughts, there is a script unfolding. You are not only bone and muscle, not only warmth and pulse. You are language. Inside you, in places too small to see, trillions of cells glow softly with activity. Each one carries a library. Each one contains a manuscript so old that it predates forests, predates birds, predates even the idea of names. This manuscript is written in an alphabet of 4 letters. Not letters of ink and paper, but letters made of chemistry: adenine, thymine, cytosine, and guanine. You do not see them. You do not feel them directly. And yet they are there, paired carefully inside every cell, arranged in long, winding strands. These letters form your DNA, deoxyribonucleic acid, a molecule so slender that thousands could fit across the width of a single hair, and yet so powerful that it guides the growth of your body from a single cell into the being you are now. Imagine that. Every freckle, every curve of your fingerprint, the color of your eyes, the architecture of your lungs, all of it emerges from combinations of 4 chemical symbols. It is as if your entire body is composed from a quiet poem written in an alphabet of 4 notes. You begin to feel the gentleness of this idea. These 4 letters are not loud. They do not shout commands. They do not glow with dramatic light. They simply pair and repeat and sequence themselves with patient precision. Adenine always pairs with thymine. Cytosine always pairs with guanine. Like partners who know one another deeply, they fit together with dependable familiarity. This pairing is not random. It is guided by shape and charge, by the subtle geometry of molecules. Each letter has a specific structure, and only its correct partner can align in just the right way. They are like puzzle pieces designed to interlock, forming rungs of a ladder that spirals gently within each cell. You carry this ladder everywhere. And yet you are not alone in carrying it. Every tree outside, every bird settling into its nest, every insect drifting in the night air carries the same 4 letters. The alphabet is universal across life on Earth. You share it with moss, with whales, with ancient bacteria that thrive in hot springs. The words may differ, the sequences may change, but the letters remain the same. There is something comforting about that. Within your cells, these letters are arranged in long sequences, like sentences written without spaces. A segment of DNA that carries a meaningful instruction is called a gene. A gene is not a thought, not exactly, but it is a recipe. It instructs the cell how to build a protein. Proteins are the workers and structures of your body. They form your hair and nails. They carry oxygen in your blood. They send signals across your neurons. They fold into intricate shapes, each shape determined by the order of those 4 letters. You can imagine it like this: the DNA is a cookbook, and each gene is a recipe. The ingredients are amino acids, small molecular building blocks. The instructions for assembling them are encoded in triplets of letters. 3 letters at a time form a word, and each word corresponds to a specific amino acid. In this way, the 4-letter alphabet expands into a vocabulary large enough to build you. It is astonishing, and yet it happens silently. At this very moment, within your cells, sections of DNA are being read. Molecular machines move along the strand, copying segments into a related molecule called RNA. This process is called transcription, but you can think of it as tracing, a gentle copying of a particular passage. The RNA then travels to tiny cellular factories called ribosomes, where the instructions are translated into proteins. Translation. The word feels soft. A language within a language. Chemistry becoming structure. Letters becoming form. You begin to notice how intimate this is. The script inside you is not distant. It is not abstract. It is actively shaping your breath, your heartbeat, your ability to think these thoughts. The firing of neurons in your brain depends on proteins built from instructions written in those 4 letters. Your immune system recognizes invaders because proteins fold in particular ways, determined by sequences encoded long ago. And yet the alphabet itself is simple. Why 4 letters? Why not 3? Why not 7? That question rests quietly for now. Tonight, you simply observe that 4 is enough. 4 allows for variation, for complexity, for the vast diversity of life. With 4 symbols arranged in different orders, nature can write endless combinations. It is like music composed from just a few notes, yet capable of infinite melodies. You are one melody. The letters in your DNA stretch for billions of pairs, coiled and folded carefully to fit inside each microscopic nucleus. If you were to unwind all the DNA in a single cell, it would extend nearly 2 meters in length. And yet it fits within a space so small you cannot see it without powerful microscopes. There is elegance in that compression. The DNA does not float freely. It wraps around proteins called histones, forming structures that resemble beads on a string. These beads coil further, organizing the long molecule into chromosomes. This organization protects the code, regulates its use, and ensures that when cells divide, each new cell receives its own complete copy of the script. Copying. Protecting. Reading. The verbs are gentle, repetitive, reassuring. You begin to sense that your body is less like a machine and more like a library in constant quiet motion. Pages are turned. Sections are referenced. Certain chapters are opened in one cell but closed in another. A skin cell reads different passages than a liver cell, even though they carry the same complete text. This selective reading is what allows your cells to specialize. The same alphabet, the same full manuscript, yet different expressions depending on context. It is as if every cell chooses which poem to recite. And you, resting here, are the sum of those recitations. There is humility in this realization. You are built from molecules that obey simple physical rules: attraction, repulsion, bonding. And yet from these rules arises awareness. From 4 letters arranged in particular sequences emerges the ability to wonder about those letters. You are language becoming conscious of itself. As you drift deeper into stillness, you feel the quiet persistence of that alphabet. It does not sleep. It continues its work. It continues to be read, copied, repaired. Small molecular guardians scan the DNA for errors, correcting mismatches to preserve the integrity of the script. Because the script matters. Without faithful copying, without careful pairing, the instructions would blur. And so life has evolved mechanisms to guard this alphabet, to maintain its clarity across generations. The letters you carry are echoes of ancient organisms that lived billions of years ago. older than mountains, older than oceans as we know them. And yet the story did not begin with letters. Before the alphabet, before the first gene was ever written, there was something simpler: chemistry alone, atoms drifting and colliding in the young Earth's restless seas. And as you rest tonight, breathing softly, you may begin to wonder how those drifting atoms first arranged themselves into symbols at all. Before there are letters, before there are cells, before there is anything that could be called a body, there is motion. You travel back in imagination to a young Earth, more than 4 billion years ago. The planet is warm, its surface restless. Volcanoes breathe thick clouds into the sky. Lightning flickers through heavy atmospheres rich with gases. The oceans are not yet calm blue mirrors but dark, mineral-laden waters, stirred by heat rising from the planet's interior. There is no life here. No eyes. No heartbeat. No alphabet. And yet there is chemistry. Atoms drift in the ancient seas: carbon, hydrogen, oxygen, nitrogen, phosphorus. These are simple elements, born in stars long before the Earth itself forms. They carry no purpose. They do not plan. They simply follow the quiet rules of physics: attraction, repulsion, balance. You imagine them as dancers without choreography. They move because they must. They collide because motion brings them together. And sometimes, when the geometry is just right, they join. A bond forms. A molecule emerges. Carbon, with its remarkable flexibility, becomes a central character. It can form 4 bonds, reaching out in different directions like a patient architect. Hydrogen attaches lightly. Oxygen brings polarity, a gentle imbalance that allows molecules to interact with water. Nitrogen introduces the possibility of rings and chains. These combinations are not rare accidents. In the warmth of early oceans, with energy supplied by sunlight, lightning, and volcanic heat, molecules constantly assemble and break apart. The process is rhythmic. Formation. Dissolution. Formation again. There is a kind of rehearsal happening. In 1953, long after these ancient oceans cool and continents settle, scientists named Stanley Miller and Harold Urey recreate a version of this early atmosphere in a glass apparatus. They circulate gases and send sparks of electricity through them. After days, they discover something astonishing: amino acids, the building blocks of proteins, form spontaneously. Chemistry, given the right conditions, begins to build complexity. You let that idea settle. The building blocks of life can arise from simple ingredients, stirred by energy. In the young oceans, similar processes unfold again and again. Amino acids form. Sugars form. Lipids, fat-like molecules with water-loving heads and water-repelling tails, gather into tiny spheres called vesicles. These vesicles create boundaries, separating inside from outside. They are not alive, but they hint at containment. Containment is important. Without boundaries, reactions diffuse into the vastness of water. With boundaries, molecules are held close together. Concentration increases. Interactions become more likely. Inside a vesicle, chemistry can become more focused, more persistent. You imagine countless microscopic spheres drifting in warm seas, each one a tiny experiment. Somewhere among these experiments, certain molecules begin to display a new property, the ability to copy aspects of themselves. Not perfectly, not with intention, but with a tendency toward repetition. This is where the story begins to lean toward language. There are molecules called nucleotides, each composed of a sugar, a phosphate group, and a nitrogen-containing base. These bases are cousins of the letters you encountered before: adenine, cytosine, guanine, and uracil. In early Earth chemistry, similar bases can form under plausible conditions. Nucleotides have a remarkable trait. They can link together into chains. The phosphate of one attaches to the sugar of another, forming a backbone. And the bases, the letter-like structures, extend outward, capable of pairing with complementary shapes. This pairing does not require awareness. It is guided by hydrogen bonds, subtle attractions between specific atoms. If two bases have matching shapes and charge distributions, they fit together more comfortably than mismatched pairs. Comfort is enough. In a world governed by probability, comfortable pairings persist slightly longer. They are more stable. And stability, even by a small margin, matters. Imagine a short chain of nucleotides forming by chance on the surface of a mineral, perhaps clay. Minerals can act as scaffolds, aligning molecules and increasing the likelihood of bonding. A chain forms. Then, free nucleotides in the water encounter it. Complementary bases attach along its length. A second chain begins to assemble, guided by the first. It is not perfect copying. It is not efficient. But it is a beginning. This hypothesis, that RNA or RNA-like molecules arise before DNA, is known as the RNA world hypothesis. RNA differs from DNA in subtle ways. It uses uracil instead of thymine. Its sugar is ribose rather than deoxyribose. It is generally less stable. But it has a gift: it can both store information and catalyze reactions. Certain RNA molecules, called ribozymes, can speed up chemical processes, including aspects of their own replication. You can feel the shift here. Chemistry is no longer only forming random structures. It is forming patterns that influence their own continuation. A strand that can assist in making copies of itself becomes more common over time, simply because it persists. There is no intention, only selection. Selection, at this stage, is gentle and statistical. Molecules that last longer accumulate. Those that break apart quickly fade. Over countless cycles of formation and dissolution, more stable and more replicative arrangements become slightly more abundant. You imagine the oceans as vast laboratories, running experiments without pause for millions of years. Eventually, networks of molecules begin to cooperate. One strand may help another replicate. Vesicles containing these networks may grow and divide when they become too large. A primitive form of heredity emerges. Contents of one vesicle are passed into two. Still not life as you know it, but closer. DNA, with its greater stability due to its double-stranded structure and deoxyribose sugar, likely evolves later. It becomes the long-term archive, while RNA handles more immediate tasks. But before DNA's quiet alphabet settles into place, chemistry must first explore its options. You consider how improbable this all might seem. And yet, given enough time, enough space, and the steady laws of physics, complexity can arise naturally. Order does not require a designer, it can emerge from repetition and constraint. Water provides a medium. Energy provides motion. Carbon provides flexibility. Minerals provide surfaces. And hydrogen bonds provide specificity. From these ingredients, patterns begin to echo. The first hint of language is not meaning. It is repetition. A sequence that can be copied becomes a rudimentary sentence. A pattern that influences its own continuation becomes a primitive story. You rest with that thought. Before letters, there is chemistry discovering what shapes are possible. Before genes, there are molecules testing which arrangements endure. The alphabet you carry tonight is the descendant of countless ancient trials, most of which dissolve quietly back into water. And as you imagine those early oceans dark, warm, filled with drifting atoms, you begin to wonder how, from all the possible chemical symbols that could have formed, for particular letters eventually came to dominate the script of life. You return now to the 4 letters, but this time you look at them more closely. Adenine, thymine, cytosine, guanine. They sound almost musical when spoken slowly. Not dramatic, not grand. Just gentle syllables, shaped by chemistry rather than poetry. And yet these 4 small molecular structures form the foundation of every tree, every coral reef, every memory you have ever held. You imagine holding them in your palm like smooth stones gathered from a riverbank. Each one slightly different in shape. Each one carrying subtle edges and curves. If you were to magnify them billions of times, you would see rings of carbon and nitrogen atoms, arranged in patterns that give each base its identity. Adenine and guanine are larger, structured as double-ringed molecules called purines. Cytosine and thymine are smaller, single-ringed pyrimidines. This difference in size is not decorative. It is essential. When DNA forms its familiar double strand, a large base must pair with a small base. If 2 large bases tried to meet, the strand would bulge. If 2 small ones paired, it would pinch inward. Stability depends on symmetry. The width of the double helix remains constant because each rung of the ladder is composed of 1 purine and 1 pyrimidine. You can feel the quiet precision in that. Adenine pairs with thymine. Guanine pairs with cytosine. No other combination is as comfortable. The atoms align in such a way that hydrogen bonds form between them, 2 bonds between adenine and thymine, 3 between guanine and cytosine. These bonds are not strong like covalent bonds that hold atoms within a molecule. They are softer, more flexible. They allow pairing without permanence. This softness matters. If the bonds were too strong, the strands could not separate during replication. If they were too weak, the structure would fall apart. The balance is subtle, like a handshake, firm enough to be reassuring, yet gentle enough to release. But still, you wonder, why these 4? Early Earth chemistry could have produced many nitrogen-containing bases. Experiments have shown that alternative bases can form under plausible prebiotic conditions. Some could, in principle, pair in similar ways. And yet life, as it evolved on this planet, settled on this particular quartet. Part of the answer lies in chemistry's quiet preferences. The canonical bases A, T, C, and G are relatively stable in water. They do not degrade too quickly under normal biological conditions. They can be synthesized from simpler molecules that plausibly existed on early Earth. And crucially, their pairing is specific enough to allow reliable copying, yet flexible enough to tolerate minor variations. Reliability is everything for heredity. Imagine an alphabet of 10 letters instead of 4. The number of possible combinations would increase dramatically. But so would the likelihood of mismatches during copying. With too many options, the probability of error rises. Information becomes harder to preserve. Now imagine only 2 letters. The system becomes simpler, but the vocabulary shrinks. With 2 symbols, sequences can vary, but the potential complexity of encoded information is limited. Proteins, which require 20 different amino acids, would be harder to specify efficiently. For letters, create a sweet spot. With 4 symbols arranged in triplets, you obtain 64 possible combinations. This is more than enough to encode 20 amino acids, with redundancy built in. That redundancy allows for resilience. A mutation in the 3rd position of a codon often does not change the resulting amino acid. The system is buffered against error. You sense the elegance in that redundancy. It is not wasteful. It is protective. But chemistry also shapes the choice more directly. The specific arrangement of hydrogen bond donors and acceptors in these bases makes mismatched pairings energetically unfavorable. Adenine does not comfortably bond with cytosine because their hydrogen bonding patterns do not align properly. Guanine does not pair well with thymine for the same reason. It is not that the wrong pairings are impossible. It is that they are less stable. And in a world of countless molecular interactions, even small differences in stability accumulate over time. Over billions of years, systems that copy more accurately outcompete those that copy less accurately. A slight improvement in fidelity can mean the difference between persistence and dissolution. So the 4 bases that happen to pair cleanly, consistently, and with the right balance of strength become favored. Selection operates even at this molecular level. You imagine countless early genetic systems experimenting with slightly different alphabets. Perhaps some used alternative bases. Perhaps some relied on slightly different backbones. Many of these systems likely vanished, leaving no trace. The one that persists is the one that balances stability, flexibility, and efficiency most gracefully. And that one uses 4 letters. There is another layer to this story. DNA is not the only possible genetic molecule. Scientists have created synthetic systems called XNAs, xenonucleic acids, with alternative backbones and bases. Some can store information and even evolve in laboratory conditions. This shows that chemistry allows multiple possibilities. And yet on Earth, life converges on this particular solution. You might think of it as path dependence. Once a system gains a foothold, once early life adopts a particular alphabet, future evolution builds upon it. Changing the fundamental letters would require rewriting every gene, every protein coding sequence, every regulatory mechanism. It becomes easier to modify words than to replace the alphabet itself. The alphabet becomes locked in. And so, for billions of years, adenine pairs with thymine. Cytosine pairs with guanine. Across continents, across extinctions, across ice ages and meteor impacts, the pairing persists. There is something quietly beautiful in that continuity. You carry molecules that echo ancient seas. The letters in your DNA are chemically similar to those in the first cells that ever formed stable lineages. The same hydrogen bonds that hold your genes together once held together the genomes of single-celled ancestors drifting in primordial oceans. It feels intimate, almost tender. When you consider the vastness of the universe, the idea that life depends on 4 specific molecular shapes seems fragile. And yet here you are, breathing, thinking, reflecting, all because these 4 shapes happen to fit together in just the right way. The choice was not made consciously. It was not selected from a menu. It emerged from constraints and probabilities, from the way atoms prefer to arrange themselves. The inevitability lies not in destiny, but in chemistry's quiet logic. You rest with the sense that these 4 symbols are both arbitrary and necessary. Other alphabets might be possible elsewhere in the cosmos. But here, on this planet, this quartet becomes the foundation. And as you hold their names softly in your mind, adenine, thymine, cytosine, guanine, you begin to picture how they assemble not just as pairs, but as a twisting structure, a spiral ladder rising gently within every cell, carrying memory in its rungs. Now you begin to see it. Not as a flat string of letters, not as scattered symbols, but as a shape, a form that curves and turns with quiet grace. Inside each of your cells, folded carefully within the nucleus, there is a structure that resembles a ladder gently twisted into a spiral staircase. This is the double helix. You imagine holding a ladder in your hands. 2 long rails run parallel to one another. Between them, rungs connect side to side. Now imagine that ladder slowly twisting, turning in a smooth spiral, like a ribbon curling in slow motion. The rails become the sugar-phosphate backbones of DNA. The rungs are the paired bases, adenine with thymine, cytosine with guanine. It is not a chaotic twist. It is precise. About 10 base pairs complete each turn of the helix. The diameter remains constant because each rung pairs a large base with a small one. The geometry is so consistent that if you could shrink yourself down to the molecular scale, you would find the spiral repeating rhythmically, like a well-composed melody. This shape was not imagined out of nothing. In 1953, James Watson and Francis Crick proposed the double helix model, building upon experimental data from Rosalind Franklin and Maurice Wilkins. Franklin's X-ray diffraction images revealed a distinctive pattern, an X-shaped shadow that hinted at a helical structure. From patterns of scattered radiation, a hidden architecture emerged. You think about that for a moment. Light passing through molecules. Shadows revealing form. Humans deciphering the shape of something too small to see directly. There is poetry in that discovery, but tonight you rest not on the drama of history, only on the structure itself. The double helix is elegant because it solves several problems at once. First, it protects information. The bases, which carry the genetic code, are tucked inside the spiral, shielded from the watery environment of the cell. The sugar-phosphate backbone faces outward, interacting with surrounding molecules. This arrangement keeps the letters safe from random chemical damage. Second, it allows copying. Because each base pairs specifically with its partner, each strand carries the information needed to reconstruct the other. If the strands separate, adenine will attract thymine, cytosine will attract guanine. The complementary nature of the strands makes replication not just possible, but logical. You can feel the quiet genius in that simplicity. The strands run in opposite directions, one from what scientists call the 5-foot end to the 3 end, the other in reverse. This antiparallel orientation ensures proper alignment of chemical bonds along the backbone. It is a subtle detail, but it contributes to the stability of the whole structure. Stability is everything here. Too rigid, and the helix would resist necessary changes. Too fragile, and it would unravel at the slightest disturbance. Instead, the double helix balances strength and flexibility. The covalent bonds along each backbone are strong, anchoring the structure. The hydrogen bonds between bases are weaker, allowing the strands to unzip when needed. Unzipping is a gentle word. During replication or transcription, enzymes move along the DNA, separating the strands just enough to read the code. The helix opens like a zipper being carefully drawn apart. After the process completes, the strands return to their paired state, the latter twisting back into its calm spiral. There is rhythm in this motion: open, read, close. Open, copy, close. You imagine the helix as a living archive, continuously accessed but rarely damaged. And yet damage does occur. Ultraviolet light from the sun can cause thymine bases next to each other to form abnormal bonds. Reactive molecules within cells can alter bases. But cells are equipped with repair mechanisms, molecular caretakers that patrol the DNA, correcting errors and restoring proper pairing. The structure itself aids these repairs. Because the strands are complementary, one can serve as a template to fix the other. If a base is altered on one strand, the matching base on the opposite strand provides guidance for correction. The latter remembers its intended shape. And this shape is not static. DNA coils and supercoils, wrapping around proteins called histones to form nucleosomes. These nucleosomes organize into higher-order structures, compacting meters of DNA into a nucleus only micrometers wide. The double helix bends and loops, creating regions that are more tightly packed and others more accessible. Accessibility matters. Genes that need to be expressed are often found in more open regions, where enzymes can reach them. Genes that are temporarily silent may be tucked away in tightly wound sections. The architecture of DNA is dynamic, responding to signals from the cell and the environment. You begin to see that the double helix is not just a storage device. It is interactive. Its shape influences which genes are active, which are quiet, which are available for transcription. The three-dimensional arrangement within the nucleus affects how distant regions of DNA can come into contact, influencing regulation. A ladder, yes, but also a landscape. If you were to stretch out all the DNA from one of your cells, it would measure roughly 2 meters in length. And yet it fits into a nucleus only about 6 micrometers across. This extraordinary packing requires precise folding. The helix loops into domains, domains fold into territories, and chromosomes occupy distinct regions within the nucleus. Each cell in your body contains this twisting ladder, folded and refolded with astonishing care. You reflect on how this structure, discovered only a few generations ago, has become one of the most recognizable symbols of life. The double helix appears in textbooks, logos, artwork. But beyond symbolism, it remains a physical reality within you, a molecule turning quietly in the darkness of your cells. It holds memory, not in the way your mind holds memories, but in the sense that it preserves sequences across time. The arrangement of bases you carry is inherited from your parents, who inherited theirs from theirs, stretching back through unbroken lineages to the earliest cells. The helix is a thread connecting you to ancient oceans. And yet, for all its elegance, the double helix is only a structure. A beautiful architecture waiting to be used. Information stored in rungs must still be read, interpreted, and transformed into the molecules that shape your body. The latter holds instructions, but it does not act alone. As you drift deeper into stillness, you imagine the strands gently parting, enzymes gliding along, copying each letter with quiet precision. Because the true power of the double helix is not only in how it holds memory, but in how faithfully it can pass that memory forward. You picture the double helix resting within the nucleus, coiled and folded. Then, gradually, specific proteins gather at particular starting points along the DNA. These starting points are called origins of replication. They are like bookmarks placed in the manuscript, marking where copying should begin. An enzyme called helicase approaches. Its role is gentle but decisive. It begins to unwind the double helix, breaking the hydrogen bonds between paired bases. The latter unzips. The spiral loosens. Two single strands begin to separate, revealing the letters that were once hidden inside. You imagine the soundless motion of this unzipping, smooth, deliberate. As the strands part, another set of proteins stabilizes them, preventing them from snapping back together. The exposed bases are vulnerable, but they are also ready. Ready to guide the construction of new complementary strands. This is where the quiet scribe enters, DNA polymerase. DNA polymerase is not conscious. It does not see the letters as symbols, but it recognizes shapes and chemical cues. Moving along one of the separated strands, it reads each base and selects the appropriate complementary nucleotide from the surrounding pool within the cell. If it encounters adenine, it adds thymine. If it sees cytosine, it adds guanine. The logic is embedded in molecular geometry. You begin to sense how elegant this is. Each original strand serves as a template. From one helix, two are formed. Each new double helix contains one old strand and one newly synthesized strand. This is called semi-conservative replication. Half old, half new. A quiet blending of past and present. But copying DNA is not as simple as moving in a straight line. DNA strands run in opposite directions, chemically speaking. DNA polymerase can only build a new strand in one direction from what scientists call the 5-foot end toward the 3 end. Because of this, replication proceeds differently on the two template strands. On one strand, called the leading strand, synthesis occurs smoothly and continuously. The polymerase follows the unwinding helicase, adding nucleotides in an unbroken sequence. On the other strand, called the lagging strand, synthesis is more complex. Because of the opposite orientation, the polymerase must work in short segments. These segments are known as Okazaki fragments, small stretches of newly synthesized DNA that are later joined together by another enzyme called ligase. You picture it as stitching fabric in sections, carefully sewing pieces into a seamless whole. Despite this complexity, replication is astonishingly accurate. The error rate of DNA polymerase is remarkably low, about 1 mistake per billion nucleotides added. And even that is not the end of the story. DNA polymerase has proofreading ability. As it adds nucleotides, it checks its work. If it detects a mismatch, it can remove the incorrect base and replace it with the correct one. You imagine the enzyme pausing, noticing a slight misfit, gently correcting it. There are also additional repair systems that scan the DNA after replication, searching for mismatches that escape immediate proofreading. These molecular guardians patrol the genome, maintaining the clarity of the script. This layered protection explains how the genetic code can persist across generations with such stability. And yet, perfection is neither possible nor entirely desirable. Occasionally, a mismatch slips through. A base is inserted incorrectly. A small segment is duplicated or deleted. These changes are called mutations. Most are neutral, occurring in regions that do not affect protein sequences. Some are harmful. A few, very rarely, may confer an advantage under certain environmental conditions. Replication, then, is both conservative and creative. It preserves the core script with high fidelity, yet allows small variations to accumulate over time. Without accurate replication, life would dissolve into chaos. Without occasional errors, evolution would stall. You begin to appreciate the balance. Inside you, right now, millions of cells are dividing. Skin cells renew. Cells lining your intestine replace themselves. Even in tissues where cells divide less frequently, DNA replication has occurred countless times since you began as a single fertilized egg. From that one original cell, containing one complete genome, trillions of copies have been made. Each cell in your body carries essentially the same DNA, replicated again and again with extraordinary care. There is something humbling about this quiet labor. Replication requires energy. It requires coordination. It requires timing. The cell cycle includes checkpoints, moments where the cell assesses whether replication has been completed correctly before proceeding to division. If significant errors are detected, the cell may halt the process or even initiate programmed cell death to prevent propagation of damage. The script is important enough to guard. You imagine the replication fork, the Y-shaped region where the DNA is actively being unwound and copied. It moves along the chromosome like a small, steady wave, leaving behind two complete helices where there was once one. Wave after wave, across the entire genome. This process is not dramatic. It is not accompanied by fanfare. It unfolds in microscopic silence, guided by enzymes that have evolved to perform their roles with remarkable precision. And yet, without this copying, there would be no growth, no healing, no reproduction. You think back to the double helix, its complementary structure making replication possible. The pairing rules you learned earlier now reveal their deeper purpose. Because adenine pairs only with thymine, and cytosine only with guanine, each strand inherently contains the information needed to recreate its partner. The structure anticipates duplication. You feel a quiet continuity here. The letters chosen long ago, shaped by early chemistry, now participate in a process that sustains you moment by moment. The unzipping and copying happening within your cells is a direct descendant of primitive replication systems in ancient oceans. And as you rest, breathing softly, you sense that replication is not merely mechanical. It is the way memory travels through time. It is how life remembers itself. But copying alone does not create muscles, thoughts, or breath. The script must be interpreted, translated into form. Letters must become instructions. Instructions must become structure. And so, as the newly formed helices settle into their spiraled calm, you begin to wonder how sequences of 4 letters transform into the living architecture of your body. Now you arrive at a quiet transformation. The letters have been copied. The double helix rests again in its spiraled calm. But the story does not end with preservation. DNA is not merely an archive sealed in darkness. It is a script meant to be read. You imagine a particular gene, a stretch of letters arranged in a precise order. Perhaps it encodes a protein used in your blood, or one that strengthens your skin, or one that helps neurons send signals across the delicate architecture of your brain. This gene is not constantly active. It waits, folded among millions of other sequences. Then a signal arrives. It might be a chemical message from another cell. It might be a developmental cue. It might be a response to temperature, light, or stress. Whatever the cause, proteins within the nucleus bind to specific regions near the gene. They loosen the surrounding DNA just enough to make it accessible. The helix opens again, not for copying this time, but for reading. An enzyme called RNA polymerase attaches to the beginning of the gene. You can think of it as a careful reader tracing a line of text. As it moves along one strand of the DNA, it assembles a complementary strand of RNA. Where DNA contains thymine, RNA uses uracil instead. The sugar backbone differs slightly, ribose instead of deoxyribose, but the pairing logic remains familiar. This process is called transcription. You imagine the gene being whispered into a new language. The DNA remains safely inside the nucleus, while the RNA copy, called messenger RNA, or mRNA, carries the instructions outward, into the broader space of the cell. The message is not yet a protein. It is still letters. But now the letters are arranged in a sequence that can be interpreted. When the mRNA reaches a ribosome, the next stage begins. Ribosomes are remarkable structures, part protein and part RNA themselves. They are molecular translators. They read the message in sets of 3 letters at a time. 3 letters form a codon. You pause here. For letters, arranged in triplets, create 64 possible codons. These codons correspond to 20 amino acids, the building blocks of proteins, as well as signals to start and stop translation. The redundancy built into this system allows for resilience. Several codons may encode the same amino acid, softening the impact of certain mutations. The ribosome moves along the mRNA, 1 codon at a time. For each codon, a transfer RNA molecule, or tRNA, brings the appropriate amino acid. Each tRNA has a small region that recognizes a specific codon and carries its matching amino acid attached at the other end. It is like a delivery system, precise and coordinated. As the ribosome advances, amino acids are linked together in a growing chain. A peptide bond forms between them. One beat after another, a string begins to lengthen. You imagine this chain emerging from the ribosome like a delicate thread. At first, it is only a linear sequence. But proteins do not remain straight for long. The chemical properties of their amino acids cause them to fold. Some regions attract water, others repel it. Some side chains form hydrogen bonds. Others create ionic interactions or disulfide bridges. The chain twists, bends, loops back on itself. It is like watching origami unfold in reverse, a flat sequence becoming a three-dimensional structure. This folding is not random. The sequence of amino acids determines the final shape. And the shape determines the function. A protein that carries oxygen in your blood, like hemoglobin, folds into a structure that can bind iron-containing heme groups. An enzyme that breaks down sugars folds into a pocket that fits its substrate precisely. Structural proteins, like collagen, form long, fibrous shapes that provide tensile strength to tissues. Function emerges from form. And form emerges from sequence. And sequence emerges from four letters arranged in particular combinations. You begin to feel the cascade. The letters do not directly create muscles or thoughts. They encode instructions for building proteins. Proteins interact with one another, forming pathways in networks. These networks create metabolism, structure, movement, sensation. Through layers of organization, complexity arises. In your neurons, proteins form channels that allow ions to flow across membranes. These flows generate electrical impulses. Electrical impulses propagate along axons, cross synapses, and influence other neurons. Patterns of activity emerge. From patterns, perception arises. From perception, awareness. You are not merely letters. You are the consequence of letters interpreted across scales. The genetic code itself, the mapping between codons and amino acids, is nearly universal among all life on Earth. A codon that specifies leucine in a bacterium will specify leucine in you. This shared code suggests a common ancestry, a deep continuity stretching back to the earliest cells. The translation machinery is ancient. And yet, each organism uses it to create something distinct. The difference between a whale and a fern lies not in the alphabet, nor in the translation rules, but in the particular sequences written in their genomes. It is like two books written with the same letters, the same grammar, but telling entirely different stories. You consider how delicate this system is. If a codon is changed, the resulting amino acid may change. Sometimes this makes no difference. Sometimes it alters folding. Sometimes it disrupts function entirely. And sometimes, rarely, it creates a subtle advantage. Meaning, in biology, is not abstract. It is embodied. A gene is meaningful because it produces a protein. A protein is meaningful because of what it does, catalyzing a reaction, forming a structure, transmitting a signal. Meaning emerges through interaction, through consequence. And yet at its foundation, there are only chemical affinities. The ribosome does not understand that it is building a muscle protein or a neurotransmitter receptor. It follows pairing rules and bonding preferences. The tRNA does not know that it carries the amino acid that will help you think or move. It simply recognizes a codon and delivers its cargo. Still, from these blind processes, coordinated complexity unfolds. You begin to appreciate how information, in the biological sense, is not separate from matter. It is embodied in molecules. The sequence of DNA is both a physical structure and a set of instructions. The two are inseparable. You were built from sentences written in molecular ink, yes, but the ink itself is alive with chemistry. As the ribosome reaches a stop codon, translation ends. The completed protein is released. It may fold spontaneously or be assisted by other proteins called chaperones. It may remain in the cytoplasm, or be transported to a membrane, or secreted outside the cell. Each protein takes its place in the larger symphony. And you, resting quietly, are the sum of countless such symphonies playing in parallel. Every breath you take depends on proteins built from codons. Every thought you think arises from networks shaped by translated genes. The letters become words. The words become structures. The structures become life. But what happens when a letter shifts? When a codon changes? When a single substitution alters the script ever so slightly? As you drift toward the edge of sleep, you begin to sense that not every copy is perfect, and that within these small changes lies the quiet poetry of evolution. Now you turn your attention to something subtle. Not the steady copying. Not the precise translation. But the small deviations that slip through. Despite all the proofreading, despite all the molecular guardians patrolling the genome, DNA replication is not perfect. Occasionally, a single letter is changed. Adenine may be replaced with guanine. Cytosine may be lost. A short segment may be duplicated or inverted. These changes are called mutations. The word can sound heavy. But tonight, you hold it gently. A mutation is simply a difference in sequence, a variation in the arrangement of the four letters. It is a small ripple in the script. Most ripples barely disturb the surface. Many mutations occur in regions of DNA that do not directly code for proteins. Some occur within genes but do not change the amino acid sequence, thanks to the redundancy of the genetic code. A codon may shift from GAA to GAG, and yet both specify the same amino acid. The protein remains unchanged. Even when a mutation alters an amino acid, the impact may be minor. Proteins are often resilient. A substitution in a non-critical region may not disrupt folding or function. The body continues, unaware. You begin to sense how buffered this system is. The four-letter alphabet, arranged in triplets, creates room for tolerance. Not every shift is catastrophic. There is space within the code for variation without collapse. But sometimes, a mutation does matter. If a substitution occurs at a crucial position, perhaps altering the shape of an enzyme's active site, or affecting the stability of a structural protein, the consequences can be significant. In some cases, the protein loses its function. In others, it gains a slightly altered one. Consider hemoglobin, the protein that carries oxygen in your blood. A single base change in the gene that encodes one of its subunits can result in sickle cell disease. The mutation alters just one amino acid, yet the shape of the protein shifts subtly. Under low oxygen conditions, the altered hemoglobin molecules stick together, distorting red blood cells into rigid, sickle shapes. One letter. One change. A cascade of effects. And yet, in certain environments where malaria is common, carrying one copy of this mutation can confer a survival advantage. The altered red blood cells make it more difficult for the malaria parasite to thrive. What appears harmful in one context becomes protective in another. You pause with that paradox. Mutation is not inherently good or bad. It is variation. The environment determines its consequence. Over generations, mutations accumulate. In organisms that reproduce, these variations can be passed to offspring. Most fade. Some persist. A few spread through populations because they offer even a slight advantage in survival or reproduction. This process is natural selection. You imagine it not as a force with intention, but as a filter. Variations arise randomly, but their persistence is not random. Those that enhance survival in a particular context are more likely to be passed on. Those that hinder survival are less likely. Across vast stretches of time, this filtering shaped species. The diversity of life you see, the differences between oak trees and orchids, between dolphins and dragonflies, arises from countless small changes layered upon one another. The alphabet remains the same. The letters are rearranged. It is like rewriting a story, one word at a time, over millions of editions. Mutation also fuels the complexity within your own body. During the development of your immune system, certain cells intentionally rearrange segments of DNA to generate a vast array of antibody molecules. This controlled genetic variation allows your immune system to recognize an almost infinite variety of pathogens. Here, variation is not an accident but a strategy. And yet, there is fragility too. Accumulated mutations in certain cells can disrupt normal regulation, leading to uncontrolled growth, what you call cancer. In these cases, the balance between stability and change is disturbed. You begin to appreciate how finely tuned this system is. Life depends on accuracy, but it also depends on flexibility. If DNA were copied with absolute perfection, if no mutation ever occurred, evolution would halt. Species would remain static, unable to adapt to changing environments. If mutation rates were too high, genetic information would degrade, and organisms would struggle to maintain functional proteins. There is a narrow window between rigidity and chaos. The four-letter system supports this balance. Its chemical pairing rules ensure high fidelity. Its redundancy allows tolerance. Its simplicity makes copying efficient. And within this framework, small changes can arise without dismantling the whole. You consider how personal this is. Every human carries millions of small genetic differences compared to any other human. Most of these differences are neutral. Some influence traits, height, metabolism, susceptibility to certain diseases. They are variations on a shared theme. You are a slightly different arrangement of the same letters. And these differences trace back through generations. A mutation that occurred in one ancestor may persist in you today. It may have arisen thousands of years ago, perhaps in response to climate, diet, or pathogens. In this way, your genome is a record of history, not just ancient history, but evolutionary history. You rest with the idea that imperfection is not merely tolerated. It is essential. Without the occasional shift in letters, there would be no adaptation, no branching of species, no resilience in the face of change. The poetry of change lies in its subtlety. A letter shifts. A codon changes. A protein folds slightly differently. A trait varies. Over time, landscapes and climates shift. Populations migrate. New niches open. Variation, filtered gently by circumstance, shapes the living world. The alphabet remains constant. The sentences evolve. As you drift deeper into stillness, you sense that these mutations do not isolate you from other life forms. They connect you. They are part of the shared process that binds all organisms in a vast family tree. Because despite the countless variations, despite billions of mutations across billions of years, the underlying code, the 4 letters, the pairing rules, the translation system remains remarkably consistent. And that consistency raises a quiet question. If variation is so common, if mutation is inevitable, why has the fundamental genetic code changed so little across all life on Earth? Now you widen your view. You are no longer focused only on your own cells, your own genes, your own quiet replication. You lift your imagination outward to forests breathing in moonlight, to plankton drifting in oceans, to bacteria dividing in warm soil. And in each of these living forms, the same 4 letters are at work. Adenine, thymine, cytosine, guanine. The same pairing rules. The same triplet codons. The same basic translation system turning RNA into protein. It is almost astonishing. A bacterium living in a volcanic vent and a human resting in a quiet room share the same genetic grammar. The code that specifies leucine in your muscle cells specifies leucine in a fern leaf. The ribosome in a yeast cell operates on the same fundamental principles as the ribosome in your neurons. You feel the intimacy of that. All life on Earth, from the simplest microbe to the largest blue whale, speaks variations of the same molecular language. The dialects differ, the sequences are distinct, but the alphabet and the core grammar remain shared. This universality is not trivial. It suggests a deep common ancestry. At some point in Earth's distant past, early life forms converged on a particular version of the genetic code. That code proved stable, adaptable, and efficient enough to persist. Once established, it became foundational. Imagine if early life had experimented with multiple incompatible genetic codes. Organisms using one code would not easily exchange genetic material with those using another. But as evolution unfolded, horizontal gene transfer, the sharing of genes between organisms, especially among microbes, became a powerful force. Bacteria can exchange plasmids carrying useful genes. Viruses can transfer genetic material between hosts. This exchange is only possible because the code is shared. A gene that encodes a protein in one organism will encode the same protein in another. The common grammar enables collaboration and competition alike. You begin to sense that universality is a kind of stabilizing force. Once a shared code spreads widely among early organisms, any major deviation becomes risky. Changing the mapping between codons and amino acids would disrupt every protein in the organism. The cost would be enormous. So the code becomes locked in. This idea is sometimes called the frozen accident hypothesis. The genetic code may have been shaped by early contingencies, particular chemical affinities, and historical events. Once it became widespread, natural selection strongly favored maintaining it, because altering it would be catastrophic. But the code is not entirely frozen. There are small variations in certain organisms. Some mitochondria, the energy-producing structures inside your cells, descendants of ancient bacteria, use slightly altered genetic codes. Certain microorganisms reinterpret a codon or two differently. These variations are rare and typically limited to specialized contexts. The core code, however, remains remarkably conserved. You reflect on how this shared language allows scientists to move genes between species. A gene from a jellyfish that produces a glowing green protein can be inserted into bacteria, plants, or even mammalian cells, and it will function. The ribosomes read it correctly. The amino acids assemble in the same order. This is possible because the code is universal. You imagine life as a vast library, where every book is written in the same alphabet. The stories differ wildly, some short, some epic, some simple, some intricate. But the letters are shared, allowing fragments to be copied, translated, and reinterpreted across contexts. It is a profound unity beneath diversity. The universality of the genetic code also suggests that all current life descended from a common ancestor, often referred to as Luca, the last universal common ancestor. Luca was not the first life form, but it was the organism from which all modern lineages descend. You do not picture Luca as grand or complex. It was likely a simple cell, living billions of years ago, perhaps in a hydrothermal environment. But within it, the genetic code had already taken a recognizable form. The same 4 letters. The same triplet codons. The same translation machinery. From that ancestor, life branched into bacteria, archaea, and eventually eukaryotes, the domain that includes you. Through countless mutations and environmental shifts, through mass extinctions and evolutionary radiations, the core code endured. Why? One reason may lie in error minimization. Studies suggest that the genetic code is structured in a way that reduces the impact of certain mutations. Codons that differ by a single base often encode amino acids with similar chemical properties. This means that a small error may substitute one amino acid for another with comparable behavior, softening the functional disruption. The code is not random. It appears optimized, at least in part, to buffer against mistakes. You consider how this connects to earlier chapters. The 4-letter alphabet allows redundancy. Redundancy allows resilience. Resilience allows continuity. Continuity allows universality. The pieces interlock. There is also something emotionally grounding in this realization. You are not isolated. At the deepest level of your biology, you share kinship with every living organism. The same molecular grammar shapes you and shapes moss clinging to stone, shapes coral reefs and hummingbirds and ancient redwoods. The language of life is shared. And yet, within that shared language, endless variation unfolds. Just as countless novels can be written with the same 26 letters of the English alphabet, countless organisms can arise from the same 4-letter genetic system. You begin to appreciate that unity does not eliminate diversity. It enables it. As you rest, breathing slowly, you sense that the genetic code is both ancient and immediate. It operates within you now, reading and translating, guiding the assembly of proteins. And it operates within bacteria in your gut, within trees outside your window, within plankton drifting in distant seas. One code. One grammar. But this raises a further question. If universality offers stability, and variation offers adaptability, What is it about the number 4, about the specific size of this alphabet, that makes this balance possible? As the thought settles gently in your mind, you begin to wonder whether 4 letters are not just historically entrenched, but mathematically and chemically well suited to the task of sustaining life. Now you turn your attention to a question that feels deceptively simple: why 4? You have seen how adenine pairs with thymine, cytosine with guanine. You have traced letters into codons, into amino acids, into proteins that shape your body. You have marveled at the universality of the code across life on Earth. And now, in this quiet hour, you consider the number itself. Why 4? Why not 2, or 6, or more? Imagine a world with only 2 letters in the genetic alphabet. Perhaps just A and T. With triplets, sequences of 3 letters, you could form only 8 combinations: AAA, AAT, ATA, ATT, TAA, TAT, TTA, TTT. That is not many. 8 codons could not adequately specify 20 amino acids, let alone start and stop signals. Life would be constrained, limited in the complexity it could generate. The diversity you now take for granted, from neurons firing in your brain to the patterns in a sunflower's spiral, would be almost impossible to encode. 2 letters are too simple. Now imagine 6 letters: A, T, C, G, plus 2 more, let's call them X and Y. The number of triplet codons would rise to 216. At first, this seems like abundance. Plenty of combinations, more than enough to cover amino acids. But here lies a hidden problem. More letters mean more possibilities, yes, but also more chance of mistakes. The cellular machinery that copies DNA must recognize each letter and pair it accurately. Each additional letter increases the risk of mismatches, of errors during replication. It becomes harder to maintain fidelity, more likely that sequences could degrade over generations. 6 letters might provide richness, but not stability. 4 letters, then, appear almost perfect. With 4, there are 64 possible codons, more than enough to encode 20 amino acids while allowing redundancy. This redundancy is elegant. Multiple codons can code for the same amino acid, providing a buffer against errors. A single nucleotide change may alter a codon, yet the amino acid remains unchanged. Life gains resilience without sacrificing efficiency. You sense the quiet logic in this balance. It is as if the alphabet was shaped by an invisible equilibrium, small enough to minimize errors, large enough to generate the complexity of proteins. Stability and flexibility coexist. Fidelity and richness harmonize. 4 letters offer enough possibilities for coding, folding, and functioning, yet maintain an elegant simplicity that preserves continuity across generations. You begin to explore the mathematical side of this choice. With 4 letters, the number of codons is 43 equals 644 caret 3 equals 6,443 is equal to 64. Of these, 61 code for amino acids, and 3 serve as signals: 1 start, 2 stops. The redundancy allows multiple codons for most amino acids. For example, leucine has 6 codons, serine has 6, while tryptophan has only 1. This uneven distribution is not a flaw, it is a safeguard. Common amino acids have multiple codons, reducing the impact of mutation. Rare amino acids have fewer codons, conserving resources. Nature builds an error correction at multiple levels. At the chemical level, complementary base pairing ensures high-fidelity replication. At the codon level, redundancy reduces functional disruptions. At the protein level, folding and interaction networks tolerate minor changes. Across scales, life protects information while remaining adaptable. You reflect on the chemical reasoning too. The 4 bases are not only the right number mathematically but also chemically compatible. Hydrogen bonding between a T and C-G pairs is strong enough to hold the helix together, yet weak enough to allow strands to separate for replication and transcription. Additional hypothetical bases might form bonds too weak or too unstable under biological conditions, or they might interfere sterically, creating bulges in the double helix. 4 letters achieve a balance between chemistry and mathematics. You imagine alternative genetic systems, like synthetic XNAs, designed in laboratories. Scientists can create 6 or 8-letter alphabets. They can even engineer new base pairing rules. These systems work in principle, showing that chemistry allows multiple solutions. Yet in natural life, evolution settled on 4. It was sufficient, reliable, and efficient enough to build the tree of life without overcomplicating the machinery. You consider the elegance in this restraint. Life does not seek maximal complexity. It seeks sufficiency. It seeks balance. Enough letters to encode information, not so many as to invite chaos. This makes you ponder evolution in a new way. It is not guided by intention, but by constraints and probabilities. The environment selects for stability and efficiency. Molecular interactions, replication fidelity, chemical affinities all shape the outcome. The number 4 is not arbitrary. It is emergent, the result of countless possibilities being tested and filtered over eons. And yet, even with 4 letters, the sequences they form are nearly infinite. Life demonstrates boundless creativity using a small alphabet. Trillions of nucleotides, folded and combined in myriad ways, produce cells, tissues, organs, and complex behaviors. The limited toolkit gives rise to almost limitless diversity. You sense the quiet poetry in that. Simplicity giving birth to complexity. Constraint giving rise to freedom. 4 letters, small and unassuming, enabling the staggering richness of life. Even at this moment, your body is interpreting these letters, folding proteins, repairing cells, generating energy, processing sensations. The quiet, rhythmic reading of 4 symbols underlies every heartbeat, every thought, every breath. And you recognize that this balance, 4 letters sufficient, yet not excessive, is repeated in other natural systems. Nature often favors equilibrium. Not too rigid, not too loose. Not too simple, not too chaotic. Life is full of such soft compromises, invisible yet essential. Finally, you begin to feel the harmony of the choice. 4 letters are enough. They are reliable. They are flexible. They carry the history of billions of years of evolution. They encode not just molecules but stories, the story of you, of the organisms around you, of life itself. And as you rest in this thought, a quiet question lingers in your mind. If 4 letters are enough to encode the building blocks of life, how does the helix hold them so delicately, balancing stability with the freedom to open, copy, and transform? Now you focus on the threads that hold everything in place. The letters you have followed so carefully, adenine, thymine, cytosine, guanine, do not float freely. They are connected, paired, and stabilized by delicate forces that seem almost imperceptible, yet their influence is profound. These are hydrogen bonds, light connections born of chemistry, and they form the quiet glue of life. You imagine each run of the double helix. One base reaching across, another answering. Two hands meeting softly in the middle, holding, but not clenching. Adenine pairs with thymine through 2 hydrogen bonds, cytosine pairs with guanine through 3. There is symmetry, predictability, a gentle order. And yet the bonds are not so rigid as to resist change. They are reversible, forgiving, flexible. It is this balance that allows the DNA ladder to perform its most essential functions. If the bonds were too weak, the helix would unravel spontaneously, strands drifting apart like sails in a storm. The letters would lose their partners, and the integrity of the script would dissolve. Proteins, enzymes, and replication machinery would struggle to interpret a constantly shifting sequence. Life would falter at its most fundamental level. But if the bonds were too strong, replication could not occur. Helicase could not unzip the strands, RNA polymerase could not read them, the flow of genetic information would stall. The code would be trapped, frozen, inaccessible. Life requires these bonds to be firm enough to maintain structure yet gentle enough to allow necessary opening and copying. You sense the quiet wisdom in this design. Nature has found a middle path. Strength paired with reversibility. Stability paired with flexibility. Life depends not on brute force, but on subtlety. The double helix responds to forces gently. As helicase moves along during replication, hydrogen bonds break one pair at a time. The strands part slowly, like curtains pulled aside to reveal a hidden stage. And when the process is complete, complementary strands naturally seek each other out, reforming the bonds and restoring the helix to its serene spiral. You imagine the cell as a room filled with tiny, invisible hands, each interacting delicately with the DNA. Enzymes smooth the path, prevent tangles, correct mismatches. And yet the bonds themselves participate, guiding pairing, enforcing rules, allowing correction. They are both passive and active, silent participants in the story of life. Hydrogen bonds are also temperature sensitive. Too hot, and they break easily, too cold, and dynamics slow. This sensitivity has consequences for evolution. Life evolved in environments where the bonds would be stable but still allow molecular motion. This balance may explain why early life favored aqueous environments with moderate temperatures, where hydrogen bonds are neither too fragile nor too rigid. You realize that the entire molecular machinery of DNA, the double helix, the complementary strands, the codons, the replication enzymes, relies on this fundamental chemistry. Everything is built upon these subtle attractions, these gentle forces that form and dissolve as needed. Even the processes you have imagined in earlier chapters, transcription, translation, mutation, all operate within the constraints and possibilities defined by these bonds. And yet, life does not always demand perfection. Hydrogen bonds can be transiently broken, altered, even mismatched. These moments create opportunities for change. Mutations occur when the wrong base pairs temporarily with another. Sometimes these mistakes are corrected immediately. Other times, they persist. From such small imperfections, evolution unfolds. Fragility enables adaptability. Gentleness permits flexibility. You reflect on the elegance of this principle. Life is neither rigid nor chaotic. It is resilient in part because the very bonds that hold it together are capable of soft yielding. They teach you a quiet lesson. Strength is not always about force, it is about balance, subtlety, and responsiveness. Proteins, too, rely on this philosophy. The same hydrogen bonds that stabilize DNA appear in alpha helices and beta sheets of proteins. The folding of chains into functional structures depends on these interactions, which are simultaneously firm enough to maintain shape and flexible enough to allow dynamic movement. Every process, every structure, emerges from a network of delicate attractions working in concert. You consider your own body. Every breath you take, every heartbeat, every thought, arises from molecules interacting with precision yet with flexibility. Life does not insist on rigidity. It thrives on bonds that are strong enough to preserve identity but light enough to permit adaptation. Even water, the medium in which all these reactions unfold, demonstrates a similar principle. Its hydrogen bonds are transient, constantly forming and breaking. This dynamic fluidity supports chemical reactions, diffusion, and energy transfer. Without these gentle bonds, life could not exist in its current form. The universality of these interactions extends beyond individual organisms. Across species, the hydrogen bonds connecting DNA strands are remarkably consistent. They are a shared molecular language, a rule written into the chemistry of life. It is another layer of stability beneath the genetic code itself. You imagine zooming in to observe them, tiny, fleeting connections flickering like candlelight across the base pairs. Invisible yet fundamental. Holding everything together, yet permitting every process to unfold. Without them, replication, transcription, translation, the entire flow of genetic information would collapse. And yet, despite their fragility, these bonds are sufficient to preserve information over billions of years. DNA sequences are transmitted from generation to generation, carrying the script of life across eons. The letters endure because the bonds are balanced, not too weak to fall apart, not too strong to resist opening. It is astonishing how such subtle forces can achieve so much. Strength through gentleness. Stability through flexibility. Persistence through impermanence. Life teaches you that the most essential structures need not be harsh or unyielding, they only need to be tuned just right. As you close your eyes, you imagine the double helix turning silently inside your cells. Every unzipping, every reformation of hydrogen bonds is a gentle rhythm, a quiet heartbeat of life itself. The letters are safe, copied, translated, yet ready for the next moment of change. And in that gentle rhythm, you sense the continuity and fragility of existence intertwined. It is a balance that has endured since the first life arose, a lesson written in chemistry long before consciousness. Tonight, as you drift toward deeper rest, you carry with you the understanding that survival often depends on gentleness, that strength is often hidden in light connections, and that life itself unfolds not through rigidity, but through a delicate equilibrium of forces. And beyond these bonds lies the next mystery, the ongoing story written and rewritten within you, still unfolding, still alive. Now you turn inward, to the living manuscript within yourself. Even at this moment, in the quiet of your body, cells are dividing, proteins are folding, and genes are being read. The script is not static. It is dynamic, continuously interpreted and reinterpreted. The story of life unfolds in real time inside you. Your genes do not act uniformly. Some remain silent, resting in the shadows of your chromosomes. Others awaken, responding to signals from your environment, your metabolism, or your own internal rhythms. Transcription factors, proteins that regulate gene expression, drift like gentle guides through the nucleus, nudging certain genes into activity, leaving others untouched. You imagine dim lights flickering across a vast library of DNA. Some shelves glow softly, some are shrouded in shadow. The genome is not a rigid blueprint but a flexible, responsive system, capable of tuning itself to meet the needs of the moment. Epigenetics adds another layer of subtlety. Chemical modifications attach to DNA or the proteins around it, influencing which genes are accessible and which remain dormant. These modifications do not alter the letters themselves but shape how the sequence is interpreted. Methyl groups, acetyl groups, small, almost invisible marks act like annotations in the margins, guiding the reading of the script without changing the underlying text. Your experiences, diet, stress, sleep, and even the air you breathe can subtly influence these epigenetic marks. In a sense, your environment is writing notes in your book, leaving quiet fingerprints that may alter how your genes express themselves. It is not magic, it is chemistry, interaction, and feedback loops unfolding over time. As you consider this, you realize that life is both remarkably stable and endlessly adaptive. The 4 letters of the genetic alphabet remain constant, preserved by the precise hydrogen bonds and robust replication mechanisms you have already explored. Yet within those letters, there is room for nuance, for interpretation, for change. Life persists, but it also responds. You visualize the interplay between DNA, RNA, proteins, and environmental cues as a vast orchestra. Each instrument contributes to the harmony, each note precise yet responsive. A gene is turned on, a protein is produced, a pathway is activated. Signals ripple through networks of molecules, adjusting and refining the activity of thousands of cells. It is a choreography of light and shadow, rhythm and pause. Even as you rest, your neurons are alive with activity. Synaptic connections adjust in response to experiences, forming the physical basis of memory and learning. Proteins synthesized from your genes are folded, transported, and integrated into structures that allow communication between cells. Your body reads, interprets, and writes its own story continuously. You begin to sense the depth of this process. The genome is both ancient and immediate. Within it lies the legacy of billions of years of evolution, sequences inherited from your ancestors, preserved through time, yet constantly interacting with the present. Each cell is a vessel carrying history, potential, and responsiveness, all in a delicate balance. Sometimes the story is rewritten in subtle ways. Mutations, which you have already encountered, introduce permanent variations. Most are silent, some minor, and some may shape your traits over time. Epigenetic marks, on the other hand, can change with your experiences, often reversible, sometimes lasting. Your own life leaves traces in the molecular text, leaving gentle imprints on the ongoing narrative. You consider the rhythm of cell cycles. Each division is a moment of renewal, a chance for the script to be read and copied anew. Errors are corrected, modifications are made, and life continues with astonishing continuity. Your cells are narrators, editors, and readers, all at once. And yet, not every page is fully understood or fully predictable. Environmental influences, stochastic variations, and complex molecular interactions introduce subtle unpredictability. This unpredictability is part of life's beauty. It ensures flexibility, allowing organisms to adapt, to evolve, to respond to change. Life is not a rigid machine but a flowing conversation between molecules, cells, and surroundings. You imagine the trillions of nucleotides inside your body, each one part of countless processes unfolding simultaneously. DNA is read, replicated, and occasionally rewritten. RNA carries instructions outward, and proteins execute them. Signals flow, pathways interact, networks adjust. Every breath you take, every thought you think, every sensation you feel is linked to this ongoing narrative. In a sense, you are reading and writing yourself continuously. Your body is both the book and the scribe, the audience and the author. The letters of the genome are unchanging, yet their interpretation is fluid, responsive, and profoundly intimate. You begin to perceive time differently. Life is not a static timeline but a continuous unfolding of molecular events. Minutes, hours, days, they are less relevant than the flow of reading, copying, interpreting, and responding. Your body moves through these processes seamlessly, often unnoticed, yet perfectly orchestrated. You reflect on the resilience of this system. Despite the constant flux, despite environmental challenges, despite the occasional errors, your genome remains largely stable. Its letters endure, its patterns preserved. And yet, within this stability lies creativity, the ability to respond, adapt, and evolve. This duality, constancy and flexibility, is a defining feature of life. As you sink deeper into calm, you consider the threads that connect you to all living things. The same processes occur in every organism, from the simplest bacterium to the tallest tree. Genes are transcribed, proteins fold, pathways interact. Life is a continuous flow of reading and writing, linking generations and species in a shared molecular story. Even the mutations, the epigenetic modifications, and the subtle variations introduced by experience are part of the narrative. They ensure that the story is never exactly the same twice. Your cells carry both the ancient legacy of evolution and the immediate marks of your existence. The script is at once historical and present, universal and personal. And in this quiet awareness, you sense a comforting truth: your life, your body, your mind, all are chapters in a story that began long before you existed and will continue long after. The four letters persist, forming sentences and paragraphs that shape your being. The story is ongoing, written and rewritten in real time. You close your eyes and feel the rhythm of the process. The quiet copying, the reading of genes, the translation into proteins, the folding, the signaling, it all flows like a gentle river within you. You are part of an unbroken chain of life, a narrative that stretches through billions of years, yet is alive in this very moment. And as your breath slows, you wonder softly, with no need for answers tonight, what future lines are quietly forming inside you, in the delicate molecular script that will shape the person you become tomorrow, the experiences you will carry, and the life yet to unfold. It is a story without end, a narrative of letters, bonds, and whispers, continuing quietly in every cell. Now you arrive at the place where your journey began, the 4 letters themselves. Adenine, thymine, cytosine, guanine. Simple shapes, small molecules, yet profound. They twist and pair in the double helix, forming the quiet scaffolding of life. You trace them in your mind, each one a note in a symphony that has played for billions of years. All the wonders you have explored, proteins folding like origami, mutations shaping evolution, universality across life, the ongoing writing within your own cells, all emerge from these 4 letters. From such simplicity arises astonishing complexity. Forests, oceans, neurons, thoughts, dreams, every expression of life is, at its heart, a rearrangement of 4 molecules. You consider the mystery. Why these 4? Why not 3, or 5, or 7? Why does life's alphabet contain just enough variety to encode the richness of existence, yet remain small enough to allow stability and fidelity? Scientists have studied this question extensively. Chemical stability, hydrogen bonding patterns, error minimization, evolutionary constraints all offer pieces of the answer. Yet the full reason remains elusive. Perhaps there is no single cause. Perhaps it is a combination of necessity, chance, and resilience. The 4 letters are sufficient to carry information, yet simple enough to be reliably replicated. They are chemically compatible, yet versatile. They provide enough codons to encode the 20 amino acids of proteins, with redundancy to buffer against error. And somehow, in this combination, life found a way to endure, diversify, and flourish. You imagine the early Earth, a swirling mixture of chemicals and warm oceans, before DNA even existed. Nucleotides formed, paired, and assembled in chains. Among countless possibilities, the chemistry favored these 4 letters. They paired predictably, they stacked efficiently, they formed strands that could open and close without collapsing. Life may have experimented with other forms, but these 4 endured. They were chosen by the constraints of chemistry and physics, by chance and by stability. And yet, every choice leaves room for variation. Even within the bounds of 4 letters, infinite sequences are possible. The letters form codons, codons form amino acids fold into proteins, proteins assemble into cells, cells form tissues and organisms interact in ecosystems. The simplicity of the alphabet belies the complexity of life that arises from it. You reflect on your own body. Inside you, 4 letters continue to be read, copied, translated. Every heartbeat, every breath, every thought is mediated through sequences of A, T, C, and G. Mutations occur silently, epigenetic marks shift subtly, environmental cues influence expression. You are both a product and a continuation of billions of years of molecular storytelling. The letters persist across all life forms. From bacteria in the soil to redwoods in ancient forests, the genetic code remains familiar and unchanged. This universality is remarkable, suggesting a single, shared origin. The last universal common ancestor, Luca, carried this code, and through countless generations, the same 4 letters continue to guide life. You feel a sense of quiet kinship with every living thing. In the smallest bacterium, in the hum of a distant whale, in the flutter of a hummingbird's wing, the same letters work, pairing, twisting, folding, and producing the machinery of life. Life's diversity rests upon a common foundation, a shared molecular language. And still, the mystery endures. Life did not have to be this way. Other arrangements of molecules could, in theory, have formed replicators or alternative genetic systems. Yet Earth settled on 4 letters. They were sufficient, efficient, and enduring. In them lies a delicate balance, stability and adaptability, simplicity and capacity, fidelity and flexibility. As you consider the scale of this wonder, you notice a rhythm. The 4 letters, the double helix, the copying, the translation, the folding, it all pulses gently, like waves on a quiet shore. You feel that rhythm inside yourself, in every cell, every breath, every thought. The code is alive in a subtle, continuous way. You allow yourself to rest in this awareness. The questions of why these 4 letters, and not others, need not be resolved tonight. The wonder is enough. The mystery is part of the beauty. Life itself is an unfolding story, written in letters, yet never fully readable, never completely predictable. In this gentle reflection, you sense the continuity of existence. Your body is reading and writing the genetic text even now. Your cells divide, proteins fold, genes switch on and off. The environment leaves faint traces. Mutations may occur silently. Your story, like all life, is ongoing, carried forward by the same 4 letters that have guided organisms for billions of years. And as your mind begins to drift towards sleep, you feel the quiet persistence of the code. It is at once stable and dynamic, ancient and immediate, personal and universal. The letters continue to pair, twist, and form sequences that encode the machinery of life. Even as your consciousness relaxes, the process continues, the double helix unwinds, RNA carries instructions outward, proteins fold into functional shapes, and cells perform their countless tasks. Life quietly proceeds, unnoticed, delicate, and resilient. The mystery remains. Why 4? Why this arrangement? Why does life work this way? Perhaps the answer will always be partly known and partly hidden, a whisper that keeps inviting curiosity and wonder. And so, you drift, carrying the knowledge that the simplest molecules can generate the most profound complexity, that life depends on balance and subtlety, and that the 4 letters continue to whisper softly within you a quiet, eternal rhythm beneath the surface of your being, guiding the ongoing story of existence.